to the series of mitotic cell cycle. In this video, we are going to discuss the actual interface, that what happens in the interface, or what happens when the plant prepares, or sorry, a cell prepares itself for division. And as you can see in this one, that it takes quite a time. It's a longer phase. This interface covers most of, like, takes most of the time of the cell cycle, and it's a very crucial phase. Here's the DNA replication take place, so it needs to occur very carefully. So very first one is that it precisely controlled sequence between one cell division and the next, because here it has a time to prepare itself. There are certain phases of it which we are going to discuss. Number one is interphase, that is G1. That is the growth phase. The S phase and the G2, again the growth phase. This S phase is the synthesis phase, in which the DNA replicates actually. So then the nuclear division, in which basically the mitosis, how the replicated DNA gets separated and they divided equally in the two daughter cells. Then the cytokinesis, that how the cell actually divides. Cytokinesis is basically the division of cytoplasm, that how it divides into two daughter cells, and it is controlled by a chemical signal which is called a cyclin. You might have heard of CDKs as well. So this is, you can say, a chemical that is controlling a cell cycle, and it needs to be controlled. It's very important. So cell cycle, or cell the cycle length may vary from one species to another. For example, if we talk about the onion root tip, it takes almost 20 hours to divide, while our intestinal cells, they take only 10 hours to divide. So it vary from species to species, cell to cell, that how do they divide. So this is the cell cycle, and we are going to discuss the very three phases: the G1 phase, in which the cell increases in size; the S phase, in which the DNA replicates; G2 phase, where the other organelles also replicate, the cell become bigger in size, and it prepares, actually it prepares cell for the mitotic division. Let's go into the detail of everyone. So first of all, G1 phase. What is G1 phase? It basically, the G indicates the growth. It's the enzymes and the proteins are made, means everything is now getting replicated. Then the S phase is the DNA replication. The two identical sister chromatids are formed. So first of all, the DNA helix, it gets separated, means a two different strands are formed, and then a new strand is formed against it. You might have heard of have learned replication in detail. If haven't, then you need to go to the video of replication to understand this concept. In G2 phase, the growth continued, the DNA checked whether the number is same, repaired, tubulin made for spindle. So a spindle fiber is very important, and we are going to learn this in mitotic phase. Okay, mitotic phase, the growth stops. Now the nucleus divided into two, and cytokinesis is in the one in which the cell split into two genetically identical daughter cells. So there is a difference in the cytokinesis of animal and plants. In animal, actually, if this is an animal cell, it actually moves inward, forming a pinch, and then it is called as cleavage furrow, and then it form like this, and then further they actually divide. But in plant cell, a plate is formed, which is formed by the vesicles. It is aligned in the center, and then it divides. So we'll go into the details of mitotic cycle in the next video. Stay tuned.